A very good morning to all my dear students. Welcome to our zoology class. And in the last class, we completed the fourth lesson. Okay. And in the last class, we studied about one important 5 mark questions, and I already told you many 2 mark and 3 mark questions also there. The genetic diseases. Okay. The genetic diseases meaning the disease caused due to the problem in the genes or chromosomes, in a single gene or single chromosome or a complete set of chromosomes. So if any abnormality takes place in this gene or in the chromosome, it leads to genetic diseases. And we studied about some examples for these genetic diseases also. And the genetic diseases are classified into, already told you, the genetic diseases are classified into two types, Mendelian disorder and chromosomal disorders. Mendelian disorders, many examples we have studied. Hemophilia, phenylketonuria, albinism, like this many examples we have studied for the Mendelian diseases. And for the chromosomal disorder also, Down syndromes, Patou syndromes, then Klinefelter syndrome. So these are the examples for the chromosomal disorder. Okay, my dear friends, I hope you all studied these question answers, and few students sent the answer to me yesterday. I corrected, then I sent them mistakes also. So really appreciate all the students those who send the answer to me. Okay students, today we are going to start the next lesson, fourth lesson, molecular genetics. It's a very big lesson. Okay, molecular genetics is a very big lesson, and in this lesson we are going to study about the genes. Okay, mainly we are going to study about the genes. Then we are going to study about the nucleic materials. Nucleic materials means the DNA, RNA, these two are called nucleic materials. So in this lesson we are going to study about genes and the nucleic materials. Understand? So first we are going to study about genes. See, one famous scientist, you all know that, Johann Gregor Mental. Okay, he is one of the famous scientists, and he only first explained the concept of these genes. Okay, he only first explained the concept of the genes, but he didn't use the name gene. Okay, simply he said one factor is present in the cells, that factor is only determining some characters in the body of the living organism. He said like that. Okay, so he didn't use any names to these factors. Okay, then another one scientist, his name is Wilhelm Jorgensen. So in the year of 1960, this Wilhelm Jorgensen only first used the term gene. So first the name gene is given by this scientist. Understand of you? And these people saying these genes only responsible for the expression of some important characters from one generation to next generation. Many phenotypic characters are controlled or expressed because of the presence of these genes. So first we are going to study about one very important 5 mark question, the properties of genes. Listen very carefully, very small question and very easy question. Listen, see, the properties of gene. The first property is numbers. Say, you all know, inside the cell, the nucleus is present. Then inside the nucleus only chromosomes are present. 46 chromosomes are present. Then inside the chromosomes only the genes are arranged in a particular order. So the number of gene is always higher than the number of chromosomes. So this is the first property of a gene. So here number, I have written the word numbers. Numbers meaning the number of gene is higher than the number of chromosome. Then the second property is arrangement. Always the genes are arranged in a linear model, in a linear structure. Then the third property is locus, meaning each and every gene is occupying a particular position in a chromosome. It is called locus. It is called locus. So the position of a gene in a particular chromosome is called locus. The next one is alleles. All the genes may exist in some alternative forms. So it is called alleles. So the alternative form of a gene is called allele. And the next property is mutation. Listen very carefully. The genes may involve in sudden change in their structure or in their composition. So sometimes the genes are involving in some changes in their structure or changes in their composition. So this is called mutation. So mutation meaning sudden change. The next one is self-duplication. See, each and every genes can produce their own copies. So this is one of the very important properties of a gene. So the genes can produce self copies. They can take their own copies. They can take their own many copies. So this character is called self-duplication. So these are the 6 important properties of a gene. The first one is numbers. Here I told the number of genes is always higher than the number of chromosomes. Second one is genes are arranged in a linear structure. Second property. Third one is locus. Every genes are occupying a particular position in a chromosome. That position is called locus. The next one is called alleles. The alternative forms of a gene is called allele. Then mutation. Mutation meaning sudden change. Sometimes the genes may involve in sudden change in their character or in their structure or in their components. Okay, so that is called mutation. And the last one is self-duplication, meaning the genes having the capacity to take their own copies. So this is called self-duplication. Understand, my dear students? So I hope you all understood this question. Properties of a gene, very important and very small question answer. Then our next topic is DNA is the genetic material. See, listen very carefully. One experiment. See, many scientists here, Crick, okay, then McLeod, like this, many biologists or many scientists believe that the proteins are the genetic material of an organism. So in maximum organism, the protein molecules are acting as a genetic materials. Okay, it was said by some scientists, some biologists. But most of the biologists or scientists believe that the DNA or the RNA is the genetic material in maximum living organism. Okay, so the Griffith, like this scientist, said that the protein molecules are the genetic material. But maximum scientists believe that the DNA and RNA is the genetic material of an organism. Understand? So here one experiment. Say Hershey, one scientist name. Then the another one scientist name is Charles. Okay, so Hershey and Charles, these two scientists togetherly they have done one experiment in the year of 1952. In the year of 1952, to prove that the DNA is the genetic material in living organisms. So to prove that DNA is the genetic material in living organism, these two scientists togetherly done an experiment in the year of 1952 by using two isotopes. One is the isotope of sulfur and second isotope is phosphorus. Okay, sulfur isotope S35, phosphorus P32. So with the help of these two isotopes, they have done one experiment to prove that the DNA is a genetic material in living organisms. Listen very carefully. See, to do this experiment, they have taken one virus and one bacterial cell. Name of the virus is called T2 bacteriophage. Here T2 bacteriophage is a virus. This virus can attack and produce many young ones inside the bacterial cell. So T2 bacteriophage is a virus. Okay, it's a virus. This virus can attack a bacteria and it can produce many young ones inside the bacterial cell. Understand? So with the help of this T2 bacteriophage virus and a bacterial cell, they have done this experiment, these two scientists. Okay, listen very carefully. They took two culture medium. This is one culture medium and this is another culture medium. In this culture medium, they added phosphorus. Okay, sorry, they added sulfur. Just imagine sulfur. So in this culture medium, they added another one isotope, phosphorus. Okay, so in this culture medium, sulfur is mixed in the culture medium. In this culture medium, phosphorus is mixed. Some T2 bacteriophage is also mixed in these two culture mediums. Understand? Then the T2 bacteriophage which is growing in the sulfur isotope is mentioned as protein. Then the T2 bacteriophage virus growing in the phosphorus isotope, it is mentioned as DNA. So listen very carefully. So to do this experiment, first they have taken two culture mediums. In one culture medium they added sulfur, and in another one culture medium they added phosphorus. And T2 bacteriophage virus also added in both culture medium. Here the sulfur containing culture medium is mentioned as protein, and this phosphorus containing culture medium is mentioned as DNA. Understand of you? Now they added one bacterial cell inside this culture medium. Immediately these two bacteriophages present in these two culture medium attacking the bacterial cells. Listen very carefully. Here already I told you these two scientists have done this experiment to prove that DNA is the genetic material. They are saying that DNA is the genetic material in maximum organisms. Understand? Say, after the infection of this T2 bacteriophage with this bacterial cell, in maximum, the phosphorus containing culture medium, already here also some T2 bacteriophage virus are mixed in this culture medium. So, these virus only closely associated or closely connected with this bacterial cell. So the T2 bacterial species, T2 bacteriophage virus present in the phosphorus containing culture medium only highly associated with the bacterial cell. These viruses are not associated with this bacterial cell. So this bacteriophage virus inserted their genetic material inside this bacterial cell, and inside this bacterial cell many bacteriophage viruses are produced. Already, what is the genetic material here? DNA. So here they mentioned DNA is the genetic material. So this T2 bacteriophage virus inserted the DNA inside the bacterial cell, and inside the bacterial cells they produce many progeny or many young ones. Okay. Finally, they but here protein. Here they mentioned protein. So these T2 bacteriophage virus present in this culture medium not associated with this bacterial cell. Okay. Here there is no infection. So here infection takes place because the T2 bacteriophage virus inserting the DNA inside the bacterial cell. Because of this reason only the bacterial cell is infected, and inside the bacterial cell many bacteriophage viruses are produced. Hope you understood. So finally they came to a conclusion that the DNA is the genetic material. This genetic material only inserted inside the bacterial cells. Hope you understood. Then one more point here. They analyze the progeny of this virus. Progeny of the virus present in the T2 bacteria, present in this culture medium, phosphorus containing culture medium. And so maximum young ones, maximum progenies carrying the phosphorus, not sulfur. The young virus, that means young T2 bacteriophage virus, not carrying the sulfur, but they are carrying the phosphorus only. So this is one of the important 5-mark question. Has here Charles experiment. So these two scientists togetherly, they have togetherly done this experiment to prove that DNA is the genetic material, and they done this experiment in the year of 1952 with the help of these two isotopes, sulfur isotope and phosphorus isotope. So to do this experiment, first they have taken two culture medium. In one culture medium sulfur is added, in another one culture medium phosphorus is added. So the sulfur containing culture medium is mentioned as protein, and the phosphorus containing culture medium is mentioned as DNA. In both culture medium, T2 bacteriophage virus are mixed. Then bacterial cell also added. After the infection, maximum the T2 bacteriophage virus growing in the phosphorus containing culture medium is closely associated with the bacteria, not this virus. Understand? So finally they came to a conclusion that the DNA is the genetic material in the living organism. Understand all of you? Understand? So it is one of the very important 5-mark question. Then next topic is chemistry of nucleic acid. Na means nucleic acid. So chemistry of nucleic acid. Listen very carefully. It is one of the very important 5-mark question. It is one of a very important 5-mark question. See chemistry. Now we are going to study about the structure of this nucleic acid. See, the nucleic acid is a very long chain-like structure, and it is made up of many subunits. So the subunit present in this nucleic acid or nucleic material is called nucleoside. Okay, so many nucleotides are present in the what? Nucleic acid. So each and every nucleic acids are made up of many subunits. That subunits are called nucleotides. The subunits are called nucleotides. Understand? And the nucleic materials has 3 important parts. Each and every nucleic material has 3 important parts. The first part is called pentose sugar. Okay, first part is called pentose sugar. And the second part is called nitrogenous base. Okay, the second part is called nitrogenous base. Understand up to? Then the third one is phosphate group. Listen very carefully. So the nucleic acid has 3 important parts. One is the pentose sugar, and the second one is nitrogenous base, and the third one is phosphate group. So first I explain about this pentose sugar. See, that based on the types of, so pentose sugar is present in all the nucleic acid, all the nucleic materials. Okay, so based on the types of pentose sugar present in the nucleic acid, the nucleic acids are classified into 2 types. One is DNA and the second one is RNA. One is DNA and the second one is RNA. So nucleic acids are classified into 2 types. One is DNA and second one is RNA. So the first point about this pentose sugar is based on the types of pentose sugar present in the nucleic acid. The nucleic acids are classified into 2 types. One is DNA and the second one is RNA. In DNA, the pentose sugar present in this DNA is called a deoxyribose sugar. Deoxyribose sugar. And the sugar present in the RNA is called ribose sugar. Okay, so deoxyribose sugar is present in the DNA and ribose sugar is present in the RNA. Understand a few? So in deoxyribose nucleic acid and ribonucleic acid, only one difference only there. Already I told you here the sugar present in this DNA is called deoxyribose sugar, and the sugar present in RNA is called ribose sugar. In deoxyribose sugar, one oxygen atom is less than the ribose sugar. So this is the one and only difference between the DNA and RNA. So once I repeat all the points about this pentose sugar. See, pentose sugar, based on the types of pentose sugar present in the nucleic material, the nucleic materials of nucleic acids are classified into 2. One is DNA and the second one is RNA. In DNA, one sugar is there, it is called deoxyribose sugar, and in RNA, one sugar is called, it is called ribose sugar. The only difference between these 2 sugars is one oxygen atom is less in deoxyribose sugar. That's all about this pentose sugar. And the second one is nitrogenous base. See, each and every DNA molecules, each and every nucleic acid has 4 important nitrogenous base. Okay, 4 important nitrogenous base. See, one is called, 2 are called purine. Okay, 2 purines and 2 pyrimidines. Okay, 2 purines nitrogenous base and 2 pyrimidine nitrogenous bases present in the nucleic acid. Okay, present in the nucleic acid. Listen very carefully. Nitrogenous base, there are 4 nitrogenous are present in nucleic acid. 2 are called purines and 2 are called what? Pyrimides. Understand? Then say adenine guanine. So adenine guanine, these 2 are the purines. Okay, adenine guanine, these 2 are the purines. Understand? Understand? Then thymine thymine, cytosine. These two are the pyrimids. These two are the pyrimids. Then one more pyrimid is also there, is called uracil. This uracil is present in RNA. Okay. So thymine is present in DNA. In RNA, instead of this thymine, one more pyrimid is there. It is called uracil. So once I repeat all these points about this nitrogenous base. In all the nucleic acid, four nitrogenous bases are there. Two are called purines and the next two are called pyrimids. The adenine and guanine, these two are the purines. Then thymine, cytosine, these two are the pyrimids. Then in RNA, instead of this thymine, uracil is present. Understand a few? Then next, phosphate group. So sugar, first parts pentose sugar over, second nitrogenous base over, and the third one is phosphate group. See, phosphate group present in a nucleic acid is derived from one acid. It is called phosphoric acid. So this phosphate group present in the DNA or RNA is derived from one acid. It is called phosphoric acid. Understand? Then the phosphate group has three important hydroxyl groups. Okay, so three hydroxyl groups are present in this phosphate group. Okay, among three hydroxyl group, two hydroxyl groups are involved in the strand formation. Then the phosphate group present in the nucleic acids only gives one of the very important property to both DNA and RNA. So the phosphate group present in DNA and RNA giving properties, one of the very important properties of the DNA and RNA. So what are the points I said about this phosphate group? So phosphate group is derived from phosphoric acid, first point, and second point, it has three hydroxyl group. Among these three hydroxyl group, two groups are involved in the strand formation, and this phosphate group is one of the very important properties of the DNA and RNA. Understand a few? Then this phosphate group forming one bond, name of the bond is called ester bond. Okay, and the last point about this phosphate group is, after the formation of one bond, phosphodiester bond, the oxygen atom present in this phosphate group is converted into negatively charged molecule. Listen very carefully. See, the phosphate group consists of one bond. It is called phosphodiester bond. So after the formation of this phosphodiester bond, the oxygen atom present in this phosphate group is converted into negatively charged. So this negatively charged phosphate group only retaining, that means keeping the nucleic materials inside the cell. It's very important to keep the nucleic material inside the cell. So I hope you all understood all these three parts of a nucleic acid. Understand, my dear students? Understand? Then next we are going to study about the nucleotides and nucleosides. Nucleoside and nucleotides. Listen very carefully. It is one of the very important 5-mark question. See, nucleoside and nucleotide. Already we have studied. Already I told you one of the important properties or parts of a nucleic acid is nitrogenous base. So this nucleoside and the structure of nucleoside and nucleotide is explained in 3 big paragraphs in your book. Okay, but the points are very small. Only 3 or 4 points only there in this topic. Listen very carefully. One nitrogenous base is linked with a sugar molecule, and immediately this nitrogenous base is converted into nucleoside. When a phosphate group is linked with the same sugar molecule, immediately this nucleoside is converted into nucleotide. So this is the first point about this topic. Say, listen very carefully. One nitrogenous base. This nitrogenous base is linked with one sugar molecule. Immediately it is converted into nucleoside. NS means nucleoside. Understand? When one phosphate is added with or linked with the same sugar, immediately this nucleoside is converted into nucleotide. This is the first point about this topic. Understand, my dear students? So when a nitrogenous base is linked with one sugar molecule, immediately it is converted into nucleoside. When one phosphate is linked with the same sugar molecule, immediately this nitrogenous nucleoside is converted into nucleotide. That's all of you. Then, second part. Many nucleotides, many nucleotides are linked togetherly and forming a chain-like structure. Forming a chain-like. So these chain-like structures are called polynucleotide chain. So listen, second point. So just imagine this is one nucleotide, this is another nucleotide, this is another nucleotide, this is another nucleotide. So many nucleotides are linked togetherly and is forming a chain-like structure. This chain-like structure is called polynucleotide chain. Hope you understood. Then, the nucleotides present in a polynucleotide chain is connected by a reaction. It is called condensation reaction. Understand all of you? So, the second point about this nucleoside and nucleotide is many nucleotide molecules are linked togetherly and forming a chain-like structure. This chain-like structure is called polynucleotide chain. And these nucleotide molecules are linked together by a reaction. This reaction is called condensation reaction. So this is the second point. And the third point about this nucleotide and nucleoside is, see, listen very carefully, the hydroxyl group present in the 3-carbon sugar of one nucleotide is linked with the phosphate present in the another one nucleotide. Then the sugar molecule present in two adjacent or two nearest nucleotides is linked by a bond called phosphodiester bond. Listen very carefully. This is one nucleotide. This is another nucleotide. Both nucleotides are adjacent nucleotides. So that means they are located nearest. Nearly located. So the OH group. So the hydroxyl group present in the 3-carbon sugar of this nucleotide is linked with the phosphate of this nucleotide. So this is one nucleotide, this is one nucleotide. So the hydroxyl group present in the 3-carbon sugar area of one nucleotide is linked or is esters with the phosphorus present in the phosphate present in the nucleotides or present in the other one nucleotides. Then this nucleotides has sugar and this nucleotides also sugar is here. These two sugars are connected by a bond. This bond is called phosphodiester bond. So hope you understood. See, the third point about this nucleoside and nucleotide is the hydroxyl group present in the 3-carbon sugar of one nucleotide is linked with the phosphate present in the nearest nucleotides. Then the sugar molecule present in two adjacent nucleotides are linked by a bond. That bond is called phosphodiester bond. This is the third point. And the fourth point about this topic is both DNA and RNA has two ends. Just imagine this is a DNA, this is a RNA. So the DNA and RNA, both nucleic acid has two ends. The one end of the nucleic acids are mentioned by using a number called 5 and the next end is mentioned by another one number 3. So in DNA, two end is there. One end is called 5 end and another one end is called 3 end. In RNA also two ends are there, 5 end and 3 end. Okay. Then in the 5 end, one phosphate group is attached, and in the 3 end, one hydroxyl group is attached. So in DNA, in the 5 end, one phosphate group is attached, and in the 3 end, one OH group is attached. Here also same. In RNA also, the 5 end phosphate group is attached, and in 3 end, hydroxyl group is present. But one difference between this DNA and RNA is, here one more OH group or hydroxyl group is attached in this, is present in this RNA, and this OH group is present in the second position. So I hope you understood this fourth point. Listen very carefully. Both the DNA and RNA has two important ends. One end is called 5 end and the second end is called 3 end. In 5 end, phosphate group is attached. In 3 end, hydroxyl group is attached. Then in RNA also, 5 end phosphate group is attached, and in the 3 end, OH group is attached. And in RNA, one more additional OH group is attached in the second position. This is the difference between these two, DNA and RNA. Understand a few? Understand a few? So these are the 4 important points. Then, now listen very carefully. See, two scientists, Wilkins and Franklin. These two scientists only first analyzed this nucleic acid with the help of X-ray. First analyzed the nucleic acid with the help of X-ray. Then two more scientists, Watson and Crick. These two scientists only first introduced a double-strand structure of DNA molecule. Listen very carefully. Wilkins and Franklin analyzed the nucleic acid with the help of the X-rays, and Watson and Crick, these two scientists togetherly introduced the double-strand structure of the nucleic acid DNA. Understand that you? Understand? Then one more scientist, Erwin Charles. Okay, so this scientist observed that the adenine is always connected with thymine. Adenine is always connected with thymine with the help of two hydrogen bonds. The help of two hydrogen bonds. Understand a few? Then the next one. Okay, the guanine is always connected with the cytosine with the help of 3 hydrogen. Listen very carefully. Erwin Charles. Okay. Okay. Erwin Charles. So this scientist observed that the adenine is always connected or linked with this thymine with the help of 2 hydrogen bond, and the guanine is always connected with cytosine with the help of 3 hydrogen bond. Understand a few? So the ratio of this adenine thymine and the ratio of guanine cytosine is always constant and equal. Okay, so suppose 10 adenine is there in a nucleic acid, 10 thymine also should be there. 10 cytosine is guanine is there, the 10 cytosine also should be there. So the ratio of the adenine thymine and guanine cytosine is always equal. Understand a few? Then, so if you know the nitrogenous base present in one strand of a nucleic acid, automatically we can predict the nitrogenous base present in the second strand. So just imagine this is one strand, this is the second strand. So in this strand, if we know the nitrogenous base present in this strand, we can predict the nitrogenous base present in the second strand. Understand a few? So that's all about this nucleoside and nucleotides. So now I said 5 important points about this nucleoside and nucleotides, and all these 5 points are explained in 3 paragraphs. So the first point, once again I repeat all these points. One, nitrogenous base. The first point in this nucleoside and nucleotide is the nitrogenous base is linked with one sugar molecule. Immediately it is converted into nucleoside. When one phosphate is linked with the same sugar molecule, immediately this nucleoside is converted into nucleotide. The second part. Nucleotides. Many nucleotides are linked togetherly by one reaction that is called condensation reaction. So all these nucleotides are togetherly linked and forming a chain. This chain is called polynucleotide chain. This is the second point. Then the third point is the hydroxyl group present in the 3-carbon sugar of one nucleotide is linked with the phosphate of the another one nucleotides. Is linked with the phosphate of another one nucleotides. Then the sugar molecules present in two adjacent or two nearest nucleotides are linked by a bond. Name of this bond is called phosphodiesters bond. And the fourth point is the DNA and RNA. Both DNA and RNA has two end. One end is called 5' and another end is called 3'. In DNA, the 5' phosphate group is connected or attached, and the 3' OH group is attached. Then in RNA also, the 5' phosphate group is there, and in 3' OH group is attached. And in RNA, one more additional OH group is present in the second position. Okay, then Wilkins and Franklin, the last point. Wilkins and Franklin, these two scientists only first analyzed this nucleic acid with the help of X-ray. Then Watson and Crick, these two people only first introduced the double strand structure of the DNA. Then Erwin Chargaff only first observed that the adenine is combined or pairing with thymine with the help of two hydrogen bonds, and the guanine is combining or pairing with the cytosine with the help of 3 hydrogen. Understand a few? So the ratio of this adenine, thymine, guanine, cytosine is always equal and constant. If you know the nitrogenous base of one strand, we can predict the nitrogenous base present in the opposite strand. Understand, my dear students? Understand a few? So today we have completed 3 important 5-mark questions, and the scientist names. These are very important 1-mark question. The 5-mark question, first we studied about the properties of genes. Properties of genes. 6 properties we studied about the genes. Then the second one is nucleotides. One experiment. Okay. So to prove that DNA is a genetic material, 2 scientists togetherly perform one experiment. Okay. So this is second 5-mark question. And the third 5-mark question gives this nucleoside and nucleotides. Okay, structure of nucleoside and nucleotides. Many 1-mark questions also there. So today itself, all try to complete today's topic. Okay, and send the answer to me. I will correct you. Okay, dear students, we can see in the next class. Thank you.